Okay, hi everyone. So I'm going to sort of continue on what Shishi Luo talked about in the previous lecture and sort of introduce some more sort of ideas into the mathematical models that we use in population genetics. So I'm going to particularly introduce demography and structure, and also introduce recombination, which will relate the haplotypes at multiple loci or positions in the genome. Okay, so to quickly recap what we saw in the last lecture, we have this simple model, this Wright-Fisher model, which is a discrete time model of mating. And as we take the population size to infinity and we rescale time suitably, we get this continuous time process called the Coalescent process. So in this continuous time process is a Markov chain which tracks the number of ancestors of a sample of size n drawn from the present, such that when the number of ancestors is k, the transition rate is to k minus 1 ancestors at rate k choose 2. So the waiting time while there are k ancestors is distributed according to an exponential random variable with rate k choose 2. And on top of this genealogical process, so this process gives you a tree at this position of the genome for the sample of size n drawn from the present. You can then impose mutations on top of this tree to get the allelic types of the sampled individuals at this position of the genome. Okay. But now you can ask, what if? So we assumed in this Wright-Fisher model that there was a constant population size, and we developed this coalescent continuum limit. But you might ask, what happens if the population size changes over time, or if there is more complicated structure in the population, in that the population is not simply randomly mating, but maybe there is some geographical dispersion of the population. Then what can you how can you extend these models? Also, you might want to ask, what if you have multiple positions of the genome that you're interested in tracking? So you're looking at the genealogical ancestry of multiple positions and then the types at those positions. And there, a very important biological process called recombination actually creates correlations in the genealogies at different positions. So we'd like to model that also. And just to sort of step back a bit and see what does all this theory that we are developing buy us, and what would we like to do with it? So sort of at the most basic level, this theory is a generative model of data, sequence data, that when you then get this data, you can try to fit parameters that you're using in this theory. And the hope is that these parameters model some kind of real the parameters of the underlying biological processes. So for example, if we model the mutation rate in this Coalescent framework or the recombination rate in this Coalescent framework, the hope is that these parameters that you use in your models have some realistic meaning. So we are interested in capturing the most important biological dynamics and mechanisms in these models with those parameters. And with demography, you'd like to infer sort of the general demographic structure of the population, which has many applications, as I'll mention in a few slides. And under this theory, you can also sort of model selection, and then you'd like to understand which regions of the genome are under selection when you're examining genetic data taken from the population. So now to get into demographic structure, we want to augment this coalescent process that we talked about in the previous lecture with demography. So as an example, this is sort of a demographic sort of peopling of the world map estimated from real genetic data. So sort of there's an estimate that the ancestors of modern humans that we see, the extant species at the moment, originated sort of in southern Africa, and around 60 to 100,000 years ago there was sort of there was a northernward migration, and then there was an out of Africa migration around 50 to 60,000 years ago. There was a wave that moved into Europe, another wave that moved into Asia, and sort of along the southern parts of Asia they further moved into Australia, and sort of the most recent migrations happened sort of via the Bering Strait bridge 50,000 years ago into sort of North America, and sort of more recently into South America. So sort of this kind of map of human demography and sort of human ancestry can be estimated from genetic data, and sort of we will use the models that I'll talk about to actually make these estimates. And in fact, that's what they did to come up with these estimates. But beyond just historical curiosity, why would we like to sort of model demography? What is the importance of this? So as you'll see in a few slides, the demographic processes that generate that underlie the population actually influence the distribution of genetic variation that you see in data. So this has obvious implications for all kinds of things, from sort of conservation genetics, where you'd maybe like to try to figure out the the population of or the number of surviving individuals of a species just by sampling from the sort of wild, and it has applications to sort of medical genetics also, where the frequency of variants, whether they are rare variants or common variants, is sort of that affects the propensity for these diseases and how correlated the diseases are with the markers in the genome. Further, when you're doing these genetic studies where you're taking the genotypic data and you're looking at whether the individual has some disease or not, and you find markers of sort of relevance which are correlated with the disease, you actually have to take into account population structure also, because you might actually find spurious correlations between the markers that you found in your tests and actually the disease, just because the frequency of the markers or these alleles is different between different subpopulations. So population structure can actually confound your association studies, your simple correlation-based studies. You'd also like to find these to get the correct null model for distribution of variation in your genome, so that maybe if you're trying to test against deviation from these null models, let's say for example your null model is that these regions of the genome are evolving neutrally, there's no selective pressure acting on them, and you don't model the demography of the region properly, and then you find that there's a deviation from predictions under your model, then you might then say that it's actually due to selection acting on those regions. But it could very well be that you didn't model the demography properly, so actually your null model was wrong to begin with. And if you had actually modeled the demography properly, you no longer may have had a significant signal for selection. So you'd like to get correct null models for doing various kinds of statistical tests. It's also of importance in forensics, where, for example, so courts look at the probability that a sample at a crime scene matches some profile from a database of individuals who were maybe convicted in the past or were suspects in investigations. And if the probability of match is low, that might be evidence for maybe further investigation into these suspects. It turns out that in these models where you compute these probabilities, it's important to actually model the demography and take that into account when calculating these probabilities. Okay, so to start off with, the simplest way to introduce demography is to have a single randomly mating population, but now the population size is no longer constant. It's no longer at equilibrium. You have some changing population size over time. So we'll start with this Wright-Fisher model where things are randomly mating, but at time t there's a population size function n of t. And now when like in the usual Wright-Fisher process, so we're going to label time, the current time would be time 0, and going back in time t would be increasing this parameter. And at generation t, you would generate nt offspring, where each offspring would pick its parent uniformly at random, independently and uniformly at random from the previous generation. Now you can sort of take the same kind of continuum limit as we did earlier with population size that didn't change in time. So we'll take sort of all these population sizes at every time to infinity at the same rate, and we'll choose a rescaling parameter so that we can get this continuous time process. So this curly n would be this rescaling parameter for time, and if you rescale time by that parameter and sort of we normalize the populations by this amount, such that this relative population size, this eta t, exists and it's some constant for every t, then we get this coalescent process now, where now that the coalescence rates in this process are no longer time inhomogeneous. They actually depend on time, because this reflects the fact that the population size is changing over time. So now when you draw a sample of size n at time 0, you can talk about the coalescence rate, and the time variable coalescence rate is now 1 over this relative population size eta t. So what this means is that if this relative population size is small, then the coalescence rates are higher. Intuitively this makes sense, because if the population size is small, 2 individuals in your sample are more likely to find an ancestor quickly, just because the population size is small. So that explains why you have this 1 over eta t. But you can get this formally by taking this limit and doing the same kind of math that Shishi talked about earlier to get the coalescent process from a constant population size.
Intuitively, what do I need about the fluctuations of population size for this kind of rescaling to?
So as I described, the population size changes in a deterministic fashion over time. You can also incorporate sort of where the population size changes randomly. There is some variability in the population size.
Oh, but it could be any pattern of population size change deterministic, and this will work.
So when you're taking this continuum limit, you're getting this this eta t function, right? So really, you have a sequence of pre-limiting models, discrete time models, that when you take the continuum limit, you get this process. And so I sort of mix both the taking the limit part of to sort of give the intuition, and also the actual construction of this continuum limit. But informally, this continuum limit, you can take any function eta t. It doesn't have to be continuous, for example. Yes.
In your Wright-Fisher graph, you have the three mergers. So what conditions do you place on the coalescences such that you only have to worry about pairs of lineages coalescing?
Right. So you would have similar conditions as in the previous talk where you had this Muller's lemma that sort of established necessity and sufficiency for convergence to the Kingman coalescent. So all this is for the Kingman coalescent, but I mean you can probably extend these lambda coalescents also, these other more general coalescents also, to handle a varying population size. Okay, so sort of to give a pictorial representation of what this variable population size coalescent process looks like. So this blue line denotes this this population size, or its scaled version. So initially the population size is large, denoting some kind of exponential growth in the population, and more further back in time there was sort of a bottleneck which denotes the fact that maybe there was a contraction in the population because maybe individuals left that population and went out somewhere else. So you sample 5 lineages or 5 individuals at the present time, and we let t and k be the waiting time while there are k ancestors for a sample of size n drawn at the present. So these are the lengths of these t nks for n equals 5. So as you can see, so you can get some intuitive sense of how variation is generated by or is affected by these changes in population size. So for example, here where the population size is larger, the coalescence rates are going to be smaller, because remember the coalescence rate is inversely proportional to the population size for any pair of individuals. So you're going to have these longer branches because individuals are taking longer to coalesce when you have this exponential growth, for example. So this also means that because you have longer branches and more proportion of the total branch lengths in this tree are being occupied by these long branches which only subtend one descendant, any mutation that occurs during this intervening times will only create a variant in one individual in your sample, right? So you'll see a lot of private mutations which only occur in one individual in your sample and not in anyone else. So this really affects the kind of variation you see in the genetic data that you sample from the population. On the other hand, when you have these bottlenecks, you have actually faster rates of coalescence, so you actually find ancestors more quickly, like there's more action going on over here. And any variation that hits these will presumably hit multiple individuals in your sample. So for example, any variation that hits this segment here will affect both individuals 1 and 2 in your sample. Variation over here will actually affect individuals 1, 2, and 3 in your sample, and so on. So these will be the more common variants that occur in your sample. And so when you have, so this is sort of how population bottlenecks can give rise to more common variants, whereas sort of more a rapid increase in population maybe recently will give rise to more rare variants. So you can try to, based on the actual kind of variance you see in data, you can actually try to infer these population size changes that occurred in the past. So this is something that people often do using data. So just sort of some quick mathematical properties or expressions that you can write. For example, the most recent, the latest coalescence event, which occurred on a sample of size n. So you can write the cumulative distribution function of this, because it's simply given by a time-varying exponential distribution. For every pair of lineages, there is this 1 over eta x rate of coalescence. So you can write the cumulative distribution function, differentiate it to get the PDF and other properties of it. But in general, these waiting times in this coalescent process are not independent random variables, because your population size changes over time. Whereas if you work with the constant size coalescent, they would actually be independent random variables. And so it's harder to get expressions for the intermediate waiting times, like the earlier waiting times in this process. But conditioning on the total amount that you've already waited in the process, it's quite easy to get the get the waiting time for the next amount. So you can write these expressions fairly easily. Okay, so far we only talked about the only structure I had introduced was a varying population size. But now you might imagine, yes.
Sorry, I was just going to ask if you could expand a bit on what you're saying about inferring population sizes from kind of the current genome.
Right. So intuitively, you got a sense that based on this population size change, this the trees. So these trees are more likely which have these skinny branches here, just because you have a very large population size here. So the trees which are actually very short are quite unlikely. So this is affecting and this affects what proportion of mutations leave signal in only one individual in your sample as opposed to a larger number of individuals in your sample. So there is a map from the population size to the frequency of variance. So for a variant that only occurs in one individual in your sample, there is some probability. Similarly for each of the other counts. So there is this map. So you might want to invert this map. Given the variance, you might want to find out what's the most likely population size change in this model that generated this data.
So if I have a particular time in the past, like 200,000 years in the past,
Yeah.
can I estimate the population size then?
Yeah, people try to do this. Yeah, they would like to. So the more data you have, probably the more you would expect to have more signal. Also here, the way I presented it was just for one. You're looking at one position in the genome, so one locus. But if you have, whole genome sequences, which you do now, from thousands of individuals, you actually get signal from. So you can think of each genomic position, the ancestry, the genealogical ancestry of these 10,000 individuals in your sample, will have some genealogical tree which might be different from the neighboring position, just because recombination breaks up correlations. So you actually have a lot more signal just because you have genomic data, and presumably you can infer what happens back in the past.
We're getting dangerously close to real data here, so maybe you can actually infer the coalescence rate that long ago.
Yeah.
Whether that has anything to do with population size, you have no idea.
So in these models, you have to scale. What we work on is this coalescence rate. So we've taken a sequence of pre-limiting models and we've taken the continuum limit, but you can get back one of the pre-limiting models that you're interested in by measuring maybe the mutation rate, the per generation physical mutation rate through biological experiments. So this gives you back the rescaling that got you into this continuum limit. But you use this continuum limit model to do the estimation of these rates, then you rescale things back using something that's outside these models.
I mean, there's also the issue that you're just estimating effective population size rather than census.
Right. So there's the other thing that we've assumed random mating. So there's a question of what that really means for a census population size. So the next models I'll talk about gives a little more structure to the population where you might have separate groups. So to move forward, you have, you could imagine now that you discretize the space where the population lives into some kind of lattice, two-dimensional lattice, where individuals migrate between the spaces they live in or the subpopulations they inhabit at some rate. So you'll have, we call these subpopulations as demes. And in deme alpha, you could imagine there's a population of size n alpha. And occasionally these demes will exchange migrants because people move around. And if you say the per generation probability that an offspring in a particular deme alpha has a parent in some deme beta going back in time, if it's given by this probability, then you can now sort of take the same kind of scaling limits that I described, that we developed earlier for the case without any structure. So what would you do? You would take each of these deme sizes to infinity. You'd take some rescaling quantity, this curly n, which would reparameterize time. And now the sample that you draw at the present will have some number of individuals from each of these demes. So it will be a tuple indexed by alpha. And now the coalescence rates for the coalescence rate for any pair, since individuals can only coalesce if they occur in the same deme, because individuals have to move around between demes. The coalescence rate for this deme alpha for any pair of individuals in that is now inversely proportional to the relative population size in that deme. Again, this makes intuitive sense. Smaller demes have smaller populations, so there's faster coalescences. Individuals in your sample find ancestors more quickly. But the most important sort of modification is that there is also migration of lineages now that occurs between demes, because these individuals are moving around. And so now the scaled parameter that represents the migration is going to be the migration rate, where it's related to the per generation, the discrete time migration rate by the scaling parameter, the rescaling of time that you used. And so really in these rescalings, you would say that you're taking this n to infinity, this curly n to infinity, while this quantity is going to 0, so that this migration rate is approaching some constant, and this is the scale migration rate that you're using. So to give concrete example of what this Markov chain looks like, when you have, say, 2 demes, we'll label them alpha and beta. So now the state of the Markov chain, the genealogical process, at time t, is given by this tuple indexed by alpha and beta. So n alpha t is the number of individuals that are ancestors of your sample that are now in deme alpha, and the number of n beta is the number of ancestors of your sample that are living in deme beta right now. So now if you look at a state n, which is n alpha and n beta, and you look at the transitions out of it in this Markov chain, this continuous time Markov chain, so now because of coalescence within deme alpha, you lose one lineage in deme alpha, so 2 individuals coalesce and find a common ancestor, and that happens at this rate, which is n alpha choose 2, which is the same rate that you have in the standard coalescent, but now it's augmented by the fact that you have to take into account the relative population size. This is a smaller deme, so this would have maybe a faster coalescence rate. Similarly, you can do this for deme beta, but sort of the new event is the migration event. So you have a migration back in time where an individual from deme alpha moves to deme beta, so you lose one individual and in deme alpha, and you gain one in individual beta in deme beta in this ancestral process, and this happens with rate proportional to this migration parameter. And since there are n alpha individuals, this is what gets multiplied for the total rate of these migrations out. And similarly, you can do the same thing for migration from deme beta to deme alpha. So how can this be visualized? So you imagine that in a one deme coalescent, you just have a tree relating the individuals. But now you have 2 demes, so the sample that you draw at present, there are 5 individuals drawn from deme 1 or deme alpha and 5 individuals from deme beta, and you can look at the ancestral tree relating the. So you can look at this ancestral. You have to draw this line now. So there's actually more structure than simply a tree relating these individuals. So here, going back in time, this individual actually found an ancestor in the previous deme, so it actually migrated. Over here, again, there was a migration that happened, and so you can represent sort of by these pictures. You would have more complicated pictures, of course, in depending on how many demes you have and the fact that you can't embed them in a plane so easily, so you won't get as nice pictures. So now, in practice, what people do to measure population structure is they like to summarize things by a single number, which gives them a sense of how structured the population is. A common measure is called FST, and to describe it, we need some notation. So if you let P subscript W of theta be the probability that 2 individuals sampled from the same deme are identical by descent. So identical by descent means that these 2 individuals had a common ancestor and there was no intervening mutation that occurred between these 2 individuals. And this was a common ancestor of these 2 individuals. So you can write this quantity for a pair of individuals sampled from the same deme. You can also write the quantity for the pair of individuals sampled from different demes. So that will be P subscript B of theta. And by the mutation process, we know that PW of theta is an expectation of this quantity e to the minus theta TW, where this is the probability that there was no mutation that occurred, where TW is the time to coalescence. So this is a random quantity now for these 2 individuals sampled from the same deme. And similarly for PB of theta, you can write the same thing. And now the way you summarize the structure in the population is by this quantity FST, which measures the difference between the probability of identity by descent within the lineage, within the same deme, minus the average probability of identity by descent, where this average means that now you're sampling 2 individuals at random, but this is across all demes. So you're taking into account the deme sizes when you're doing the sampling. And you normalize it by this quantity. The normalization would ensure that this FST is going to be between 0 and 1. 0 means that there's actually no substructure in the population. That is, this is perfectly randomly mating. So this is like 1 deme essentially, and all the this is just as in the regular coalescent without any substructure. And FST1 means that these populations are completely isolated. They don't exchange migrates between each other. So this number sort of is, gives you a convenient way of summarizing how much structure there is in the population. And for small mutation rates, this is roughly given by 1 minus the ratio between the coalescence time for two individuals sampled within a single deme with respect to the average coalescence time for any pair of individuals sampled across these demes. So various studies try to estimate this quantity FST from using existing genetic data, and they would like to sort of get a sense of how related individuals are across different parts of the world. So for humans, some of these estimate that the FST is on the order of 0.1 or 10%. And this sort of says that there's fairly little structure, that is, people across different demes or different parts of the world are fairly similar. The genetic variation across them is fairly similar. We can also sort of go into some details for very simple examples. In general, sort of working with these models with very complicated migration structures is a little tricky. You have to do Monte Carlo integration, or you have to do simulations, important sampling, other things. But for very simple examples, you can actually write out recurrences for these quantities that I described earlier. So suppose the most toy example would be you had the symmetric island model where there are these islands where you can move between any two demes, and sort of your migrations are with equal probability between any pair of demes. So if you let this migration rate be sort of m over g minus 1, where you had g demes, and you had this mutation rate, then by conditioning on what happens, what is the most recent event for a sample of size 2, whether it happened to be a migration or whether it happened to be a coalescence, you can write out, or it happened to be a mutation, you can write out the probabilities of IBD, or the probability that two individuals have a common ancestor and there's no intervening mutation between the common ancestor and these individuals. And you can solve these recurrences to get explicit values for these probabilities. And then you can compute this FST and other quantities for these simple examples. Okay, so you can also have sort of more complicated structure or more complicated demographic models where you incorporate migration and changing population sizes at the same time. So really, now when you have data, you actually look at very complicated models where you have many different populations. So sort of these big trunks denote separate populations. So there was one randomly mating population that may be split up into two at this time. Then maybe this population underwent a bottleneck, and this population underwent further splits. So you could imagine maybe this was the African population, and there was a split, and this was maybe the Eurasian population, and then there was a further split. So this might be the European, and this might be the Asian population, and maybe there's more recent migration that happens between these populations. So you could infer all these parameters based on the genetic data that you sample from these different populations at the current time. Okay, now I will switch to sort of another evolutionary force, recombination, which is very critical when you want to model sequence data. So what does recombination do? It generates new genetic variation in sexual organisms. So, as you all know, humans are diploid organisms. We have two copies of every chromosome. So these are called homologous copies or homologous chromosomes, one from the mother and one from the father. Now, in the first phase of meiosis, the synthesis phase of meiosis, what happens is each chromosome gets duplicated. It has twice, and these duplicates are called chromatids, and these sister chromatids are tied together at the centromere. So initially, this chromosome had an A allele at this position and a B allele at this position, and when it duplicated, it formed this other sister chromatid, and they were joined together at the centromere. And so this might be the maternal copy, and this is the paternal copy, or the other homologous copy of that chromosome. Now, in one of the later phases, the prophase 1 stage of meiosis, what happens is these two homologous copies come together in physical contact, and they exchange genetic material. So this sister chromatid on this copy exchange genetic material with this sister chromatid on this copy, and basically this creates a new. So now if you look at this new chromatid, it actually has a different variant from either of the paternal or maternal copies. So this creates a new haplotype or a new daughter cell copy. So at the end of meiosis, basically you started with this diploid cell and you got 4 haploid cells. So these are the copies in the 4 cells. So you have these this particular haplotype or variant was present in your maternal copy. This particular one was present in your paternal copy, but these mosaics were not present in either of the copies. So recombination has managed to generate a new haplotypic variant from existing variation. So why is it important? So as we just saw, I mean, this kind of mixing, this kind of shuffling together creates new variation. This also breaks down the correlation that is in the genealogies of the 2 different positions. So now if you look at, if this member was present in your sample, this particular allele was, or this particular position was inherited from maybe the maternal copy, and this particular position was inherited from the paternal copy. So the genealogies for these 2 positions is different. And this fact has implications for many problems, many data analytic problems in population genetics. So one of the things you want to do is phase genotype data into haplotype data. So what happens is when you sequence, you get sort of the total variation in both the maternal and paternal copies, and you like to separate this out between the 2. So you'd like to know which variants occur together on the maternal copy and which variants occur on the paternal copy. But maybe your sequencing only gives you actually a mosaic of the 2. You'd also like to use this kind of the recombination maps to impute or figure out what are the missing variations at different positions. Maybe you have information about the adjacent nucleotides, but you don't have information about this nucleotide, maybe due to sequencing error or other problems. And if you have some reference panel which tells you sort of, if you have a variant A at this position, you are most likely to have a variant B at this position, at this adjacent position. Because you have these statistics from reference data, and these 2 individuals are close together, most likely in the position you're looking at where you have missing data, just by looking at an adjacent site and looking at how the variation is distributed in your panel, you can probably guess and give some posterior estimates of what the allele would be that's missing. And similarly for disease gene mapping, disease association mapping, where you might not have full genome sequence data, maybe you only have your polymorphism data at different positions that are a subset of all the variants that occur. But because there is correlation between nucleotides that are close by together, if you can actually use the true, so maybe the true genetic marker that's correlated with the disease has not been sampled in your study, but something close to it has been sampled. So just because of this fact that recombination breaks up things at a distance away, the ancestry of these 2 markers that are close by is shared, and this will give you some idea about this. This can still help you find the rough genomic regions where the genotype is correlated with the phenotype. And similarly, you can, using this fact, you can also infer the ancestry of sort of admixed individuals at different positions of their genome. You can also use this for detecting signatures of natural selection, where maybe some allele that's selected for has some other alleles close by to it, and just because those alleles are close to it and this allele is selected and it increases in frequency, and the fact that the other allele is close by, this will actually also increase in frequency, even though this allele might not confer any selective advantage. So it sort of hitchhikes along, it freeloads along with this allele that's beneficial. And this is again due to the fact that positions that are close by are correlated to each other. So you'd like to sort of introduce recombination, so we can start with the Wright-Fisher model with recombination. So you start again with the population size n individuals. So let's start with 2 positions now, 2 loci, where there's some recombination rate or distance between them. So with probability r, which is sort of going to indicate the probability of a recombination event, these 2 loci will pick parents from the previous generation independently and uniformly at random. So for example, there was a recombination that occurred here between these 2 loci. It just so happened that these 2 picked the same parent from the previous generation. But if you looked at this particular individual, the left locus picked a parent which is different from the parent picked by the right locus. Whereas the other ones, there was no recombination, so with probability 1 minus r, you just pick a parent for both loci uniformly at random. So again, you can take the continuum limit for these things, and you can get a sort of process, a continuous time process that creates genealogies at both positions. But not just creating genealogies at both positions, it also gives you correlation between the genealogies of the two positions. So you need a more complex structure than simply a tree at either position, but you need some kind of structure that represents the joint trees at these two positions. So what are some desiderata or things we'd like from such a process? So we want to model the correlations at both loci. Marginally, though, we'd like the process at each locus to be the same process that we had developed earlier, right? This, for consistency, we don't want to have something completely new. We want something that embeds the earlier one locus processes. So, you can take sort of the extreme cases. If there was no recombination between these two positions, the genealogies at both positions should be identical because these two positions are inherited together as a unit because there's no recombination. On the other hand, if there was free recombination, like these individuals, these two positions were completely unrelated or completely uncorrelated, then you expect the genealogies to be independent. And in general, the genealogies of these two positions will have some non-trivial correlation between them because the recombination rate is something finite between 0 and infinity for these between these two positions that are located on the same chromosome. So the structure that we will develop is this ancestral recombination graph. So we start with a sample of size 4 individuals specified at 2 positions. Now the kind of events that can happen in this graph or in this structure is there could be a coalescence event as usual or a recombination event. So a coalescence event is where these two individuals, for example, find a common ancestor at both positions. But this individual going back in time experienced a recombination where the left, so this ancestor here contributed the left fragment to, or the left locus A to this individual at the present in the sample. Whereas it didn't contribute the B locus to the individual in the sample, so we actually don't we don't show it there because the ancestry of this locus is not important because it doesn't contribute any genetic material to the sample at present. And this ancestor here contributed the B locus to the sample at present, and it didn't contribute the A locus. Now going back in time, there was a coalescence event between these two individuals. So there was an ancestor here that contributed the left and right locus to this individual, contributed the left locus here, contributed the right locus to this individual here, but this didn't really matter because it didn't contribute anything to the sample at present. So you have this complicated graph structure that represents both coalescences and recombinations. Now if you look just at what's happening at the left locus for every individual in your sample at present, then you get a marginal, so marginalizing what's happening at the left locus, you get a tree just on the left or the A locus, and this is how the ancestry looks like. So we just care about what's happening at the left locus, so we don't actually trace back what's happening at the right locus. And you see that the most recent common ancestor of the samples at the left locus is actually this individual here at this locus. Whereas if you look at what's happening at the B locus, you have another tree actually, and the most recent common ancestor of the sample at this locus B is actually much higher up in the tree. So as you can see, the time spent in this process to the most recent common ancestor at the left locus is different from what it is at the right locus in this particular example genealogy. And in general, these quantities are different. So how do we actually construct this graph representation, and sort of how do we see that this graph representation generates the one locus trees consistently when you marginalize the tree on the left locus or on the right locus? Okay, so like we said, the per generation per individual probability of recombination between the two loci is R, and we can introduce the scaled version of the recombination rate, just like we had scaled versions of the mutation rate, the scaled versions of the migration rate. We have a scaled version of the recombination rate, where you take R to 0, N to infinity, and then while this approach is a constant. And now the configuration that you give for your sample is now specified by actually four numbers. So at time t, the ancestral process Markov chain has state given by this tuple. the first component of this tuple, A of t, is the number of ancestors at this time that contribute genetic material to the original sample that you sampled at time 0 at the left locus, at locus A. Similarly, B of t is the number of ancestors that contribute genetic material to the right locus in your sample. C of t will be those ancestors that contribute genetic material to the original sample at time 0 at both the left and the right locus. And D of t will be the number of ancestors at time t that don't contribute genetic material at either of the loci to your sample. So now once you write this, you can, once you create the state space, you can write this continuous time Markov chain again by going from this discrete time Wright-Fisher model that I described earlier, taking the continuum limit in the same way. And you can write these rates out. So for example, so I use stars to denote the fact that at this locus we don't have ancestral material being contributed to the sample at present. So here, for example, if you have two individuals that are trying to coalesce, which are contributing ancestral material at both loci, the transition rates from this tuple, so now you have a decrease in the C component, and the transitions happen at rate C choose 2. Similarly, you can write the transition rates that are happening for coalescence events for all of these pairs of coalescences that occur. There are sort of 10 possibilities, and you can write down all these things. You can also write what's happening for the recombination events. So here, there's a recombination event where this individual who is contributing genetic material at both loci A and B, there's a recombination backwards in time. So you have this left fragment, and the right fragment of this ancestor is not contributing any genetic material to this individual. And similarly, you have this, you have the other ancestor, which is only contributing the right fragment and not the left fragment. That's why, again, your tuple is changing. So you have an increase in the number of ancestors that are only contributing the left fragment. The ancestor that's contributing the right fragment increases by 1. The total number of ancestors surviving that contribute both fragments decrease by 1, because that's what happened here. And the other component doesn't change. And similarly, you can write the recombination rates for the other configurations also, and there are 4 such events. So you can summarize all these transitions.
Can you show why we need to keep track of D?
I'm just getting to that actually. So if you summarize the transitions out of this tuple, this state, this 4 tuple, you can write down these rates. And now the absorbing states of this Markov chain, this ancestral process, are those tuples where you've found a common ancestor that's contributing genetic material at both loci to your sample. So there's only one such ancestor when that ancestor is remaining, and you don't care about the number of individuals that are not contributing any genetic material at any loci to your sample. So these are the absorbing states of your Markov chain. And as usual, you can impose mutations on this genealogical process. Once you have this genealogical structure, this graph relating the sample, and so you have these marginal trees, you can just impose the mutations on each of these marginal trees as usual in the standard coalescent way. Now, as Daniel mentioned, this 4th component that we kept track of, the number of ancestors that do not contribute genetic material at either loci, those don't really matter, because since they're not contributing any genetic material, any mutation that occurs at those individuals don't affect the sample at present. So they do not affect the probability distribution of data you observe at present. So we actually don't need to keep track of them in the Markov chain. So now the reduced state representation in your reduced state space Markov chain has just 3 components. So we just drop the 4th component. And you can write down the transition rates again, just ignoring any transitions that sort of alter D. And now there's only one absorbing state. It's that state where you found a common ancestor that is contributing genetic material at both positions of your at both loci. So looking at this, looking at the actual states in this example genealogy that we sampled for these 4 individuals at 2 loci. So here originally my sample had 4 individuals, and this corresponds to the C component being 4, because these 4 individuals were the ancestors trivially of the sample at each of those 2 positions, each of those 2 loci. Going back in time, the first event was a coalescence event, so there is one less ancestor that contributes genetic material at both loci. Going further back in time, the next event is a recombination event, so there is now one ancestral lineage that contributes genetic material only at the left locus, one that contributes only at the right locus, and the total number of ancestors that contribute genetic material at both loci has dropped by 1, so it's 2. And so on. You can go back in this process, and eventually you find a ancestor, the most recent common ancestor at both loci, so it has contributed genetic material at both loci, and this is the first such ancestor that has done. So even though the first ancestor that has contributed genetic material at only the left locus is actually this individual. So these marginal ancestors, most recent common ancestors are different, and then the grand most recent common ancestor at both positions is different. Okay, how do we see that this process is consistent with what we saw earlier at just one genomic position? So you can actually show that the coalescent process at just locus A is embedded, the Markov chain for this coalescent process is embedded in this coalescent process with recombination at both loci A and B. So, for the state ABC in this coalescent process with recombination, there were A plus C ancestors that contribute genetic material at locus A, right? Because C contributes genetic material at both locus A and B. So we're just summing these two up. And now if you group these states together into sort of one superstate, so if you let S sub M be the set of states in this Markov chain in the recombination process, which have M sort of ancestors that contribute genetic material to your sample, where now M is the sum of A plus C. So there are actually many such states that form part of the superstate. And then you just look at transitions from the state ABC to those where this quantity, A plus C, changes. Now this is going to represent changes that move out of the superstate that you're looking at, right? And now if you look at the total rate of transitions out of the superstate, it's the sum of those transitions is simply given by M choose 2, where M was the number of individuals that contributed material at the left position. So this M choose 2 is the same rate that you see in the coalescent process, in the one locus coalescent process, right, on M, because now the state really you're looking at is with M individuals at the left locus. Okay, so this is how we see that, at the left locus it's consistent. Similarly, you can show at the right locus it's also consistent. So what are some challenges associated with modeling recombination? So this was simply for 2 loci, but you can extend this process analogously for multiple loci. So if you have, in general, k loci with some recombination distance between each pair of adjacent loci, the state space of your Markov chain, this reduced state space Markov chain, will now have 2 to the k minus 1 components in your tuple, right? Because this is going to represent all the combinations of number of ancestors that contribute genetic material at this combination of or subsets of loci that you're interested in for your sample at present. And sort of the state space itself of the Markov chain, if you sample n individuals at the present, is going to grow as n to the 2k roughly, right? So this is an enormous state space if k is large. And in fact, even for large k and realistic recombination rates, or say larger recombination rates that you see in Drosophila and some other species, this will create many sort of these fragments that are ancestral only at some subset of the loci, but not all the positions. So you're going to have the coalescent process that you have to model. You have to go back in time to simulate. You would have to go further back in time to simulate, just because the recombination rate is large and you're tracking many loci. So simulating data under these processes is very challenging. And now you can ask actually the question of actually computing the probability of some observed data. So when you have observed data, you need to integrate over the uncertainty in the genealogies at every of these positions, this joint structure in the mutations that could have happened that produce the data at present. So that becomes really challenging. And in practice, even if you just look at 2 locus data, that is, you're just modeling 2 positions at a time, and you just have maybe 2 alleles at each position, and even a small sample size of a few hundred haplotypes, computing the probability exactly of the data under this generative model is actually very difficult. And sort of many approximation or heuristics have to be used to tackle this problem. So far I haven't talked about natural selection at all, but you can sort of very easily imagine writing a Wright-Fisher model for natural selection, where the number of offsprings produced by each individual now depends on the type of the individual, because the type determines its fitness. And you can also construct a coalescent time model, which is this sort of backwards in time model for a sample. But that genealogical structure that you get from it is far more complicated than any of the things we've seen so far. And you get something called the ancestral selection graph from it. And people don't really use this for algorithmic inference from data. So a better way to handle sort of selection is through forward in time models, that is taking the forward in time continuum limits of these Wright-Fisher models. And Steve is going to talk about that in the next lecture. And yeah, this is all I had planned to talk about with extensions to the standard one locus coalescent.